Hello and welcome to this presentation on the recombinant hepatitis B vaccine. Hepatitis B is a viral infection that occurs in the liver caused by the hepatitis B virus or HBV. Its symptoms range from vomiting, inflammation, jaundice, which is the yellowing of the skin, and rarely death. It can cause cancer such as liver cancer. Contact by infected blood and other bodily fluids such as saliva can lead to an individual getting hepatitis B. The structure of HBV consists of DNA and DNA polymerase, the core antigen that surrounds it, and the surface antigen, which is the outer layer of the virus. In 1968, Dr. Baruch Blumberg first isolated the hepatitis B surface antigen, or HBsAg. A year later, working together with Millman, he developed the first vaccine for HBV, which was just a heat-treated form of the virus. Baruch later won a Nobel Prize for his research. Then in the late 70s, Maurice Hilleman hypothesized that injecting HBsAg into patients could help cure them of HBV. Finally, in 1986, Pablo Valenzuela of Chiron Corporation successfully made a recombinant hepatitis B vaccine by genetic transformation. This is the vaccine still in use today. The recombinant hepatitis B vaccine is the current vaccine used to treat HBR. The production process of the recombinant hepatitis B vaccine starts with the isolation of a plasmid from bacterium. After that, mixing it with restriction enzymes cuts the plasmid in two places. Then, a gene that codes for HBsAg is isolated from HBV and inserted into plasmid to form a cloning vector through DNA ligation. The plasmid is then inserted into Baker's yeast or Saccharomyces cerevisiae to genetically transform the organism. This cell is an ideal site for the plasmid because it accepts the cloning vector without exhibiting any uncanny changes. Afterwards, it is grown in yeast extract or YEHD medium for a few hours. This is so that the yeast cells can ferment the sugars in the extract. However, instead of producing acids, it produces hepatitis B surface antigen because of the HBsAg gene inserted into the yeast cell's DNA. This is an efficient way to produce a large amount of the vaccine without much hard work. Afterwards, the cells are washed and harvested. The next step is purification or the removal of foreign particles, which can be achieved through diafiltration, a dilution process, or size exclusion chromatography, which separates molecules by size. After this, the vaccine can be used to treat patients who have hepatitis B. From this diagram, you can see the various genes on the genetically transformed pea-glow plasmid. Each color represents a different gene on this diagram. We utilize the plasmid by making the genetically transformed bacteria develop a resistance to ampicillin, a substance which inhibited the growth of bacteria through the BLA gene. We also utilize the plasmid through the GFP gene, which signaled the production of protein that emitted green fluorescent light after being turned on by the gene Ara-C through arabinose. This lab was similar to the production process of the hepatitis B vaccine because both methods involved inserting a genetically modified plasmid into an organism in order to produce material it would otherwise not naturally produce. The bacteria in the pea-glow lab began emitting photons when exposed to arabinose because of the GFP gene, and the yeast cells began producing hepatitis B surface antigens because of a hepatitis B surface antigen gene. In other words, they both implicate modifying genomes and using recombinant DNA technology. The pea-glow lab and the manufacturing process of the recombinant hepatitis B vaccine both use gene control to their advantage. The yeast cells only produce HBsAg when exposed to sugars such as the ones in the yeast extract, and bacteria in the pea-glow lab only produce light fluorescent proteins when exposed to arabinose. The hepatitis B vaccine is the first vaccine to prevent cancer, specifically liver cancer. There are currently no fully supported side effects from taking this vaccine. In fact, taking this vaccine gives a patient indefinite protection against hepatitis B. I'd recommend the recombinant hepatitis B vaccine to anyone who currently has this disease. Thank you for watching.